remember that's DNA pol delta, looks like the S, and DNA pol epsilon looks like the E. The DNA pol S is for synthesis, so that one synthesizes as it replicates, and then DNA pol epsilon looks like the E, that's the editor that will edit the genome as it makes mistakes. Yeah.
What part did you say is interphase?
Interphase right here.
Oh, okay. Just everything except
everything that's not mitosis.
Mitosis, cytokinesis.
Yeah. Yep.
Okay.
So G1, S, G2. Here's mitosis. Here's cytokinesis. Okay, which is just the cells actually splitting. Here's just what I just said. There's stages of mitosis are PMAT, prophase, metaphase, anaphase, telophase. This is just organization and alignment of the chromosomes to be able to split into two different cells, which is a bigger task than you might think it is. The chromosome is huge. So to be able to organize the chromosome into unique structures and to be able to pull one side to the other side is actually a huge feat. And so they start condensing and aligning, and we look at metaphase a lot. This actually, what we're looking at, I don't have a, oh here, at a karyotype, you're actually looking at metaphase because they're so condensed. They're hard to look at. They're so, it looks like an unorganized mess. But what the point of mitosis is organization. So those chromosomes are then organized into, in mitosis, they're organized along the metaphase plate. And so when it's already gone through G1, it's already gone through synthesis, it's already gone through G2, so it's gone all the way through interphase. Now it's going to go through PMAT. So part of PMAT is metaphase, and so they'll line up along the middle, and they're ready to split apart. So then they'll tie up to the centrioles. The centrioles are going to form these protein structures that will pull the chromosomes apart by the centromere. So it'll pull it apart. Now you have two diploid cells that are match the original cell. So when we draw a chromosome like this, just a side note, don't forget that little strand right there is a double-stranded DNA strand. There are two strands in here that are wrapped around each other, of A's, T's, G's, and C's wrapped around each other. That's all they are. It's just a bunch of A's, T's, G's, and C's, and that's it. So they're aligned here. Now you have two. That's the process of mitosis. Sometimes it's called asexual reproduction. We don't really call it that in our cells because it's not really asexual reproduction. We call it the cell cycle. But a organism that can do this is an asexually reproducing organism, and sometimes it's called binary fission. So then we go into meiosis, which is specific to sexually reproducing organisms that need gametes to reproduce, that have gametes, that are able to utilize gametes to mix DNA. And remember, the more genetic variety, the more advantageous it is for the offspring. So here we have an organism with 6, same here, but this cell specifically is going to make either sperm or egg or spores or whatever it is, this cell specifically. So in the human, this would be one of the cells that are going to make eggs in the fetus. So in the fetus is when all females get every egg that they're ever going to have their whole entire life. So they go through this whole process almost all the way in utero. But for males, this happens 1,500 times per second. So this cell is like a stem cell. This is just like the beginning cell. And this cell is going to make sperm, eggs, spores, whatever those gametes are, those sex cells. So here they're going to align in metaphase 1. So here you can tell, here's the difference. They don't line up all the way along. They line up in tetrads. So there's 4. 1, 2, 3, 4. 1, 2, 3, 4. So what I was telling the class before maybe some of you guys got here was, if you just think 6, 6, 6, 3, 3, 3, 3. that helps you. Just write it out. Here's 6, 6, 6, 6, 6. So whatever the number of the organism it is, if it helps you just keep the numbers straight. So here you have 6 on each side. Now in metaphase 2, they're gonna line up like they normally should. And now they're gonna split apart, and now you have 4 gametes, each that are haploid, that are half the number of chromosomes. I'm just putting some terms up here. Okay, so let's run through just a couple more quick things. So we talked about yeasts versus molds. Yeasts look like this, molds look like this. Yeasts are unicellular, molds are multicellular. Yeasts can form this something called pseudohyphae. Molds can form true hyphae, or sometimes it's called mycelium when it's those branched off a bunch of hyphae together. A dimorphic fungi will change depending on temperature. So there'll be yeast at one temperature and mold at a different temperature. I'll show you a picture of that. So here's the cell wall structure of yeasts and molds. Same. They have, oh, I forgot to add ergosterol because that is going to come into play. So here is this inner membrane. Here is this inner cytoplasmic membrane. And in this cytoplasmic membrane, they have what's called ergosterol, which is very similar to all our cholesterol. It is unique. Cholesterol. It is unique to fungi and yeast, to its all fungi, molds and yeasts, but it's similar to ours. So here's ranoproteins, here's glucans, here's chitin. When you have those 3 together, it makes them unique, but really you could look, because chitin, you would know if your patient had remnants of chitin in their bloodstream, you know it's a fungal infection. They don't have an arachnid or something in there. So you know it's a fungal infection. Glucans are unique to fungi and plants, and then manoproteins are unique to fungi. So that's their structure. There's ergosterol. This is their inner cytoplasmic membrane. Here's the chitin, glucans, manoproteins. Here are the internal structures of a yeast. This is when they're budding and asexually reproducing. Here's the nucleus, nucleolus, the vacuoles, mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum. All these polka dots are ribosomes. So same with an animal cell, but we focus on yeast and molds. So colony morphology, so they look really weird on an agar plate. If you noticed an agar plate of yeast versus an agar plate of bacteria, you'd be like, what is that? That doesn't look normal. So they're like creamy and heavy, there's a ton of it. So they're different, and they require a lot of glucose in the medium. Under the microscope, they look like spaghetti and meatballs. Sometimes they just look like the meatballs themselves. Yeast, sometimes they don't have pseudohyphae, so it's just something to think about. So molds, they have the hyphae, mycelium, we talked about that. They have the same structures as yeast, except they also have a Spitzenkorper that will dig into rigid structures for growth, which is bad for our patients. The only difference between our other organisms internally are they have these ergosterol crystals. I don't really care if you know that. Yeah?
What about the cell walls? Are they the same materials, or?
The cell wall between a yeast and a mold is the same.
Okay, they're both.
Yes,
okay.
same. Yes. Okay, so here's vegetative hyphae and aerial hyphae. So we worry about vegetative hyphae because that's how the cytopathic effects digs into the tissues. We worry about aerial hyphae because that's what creates the fruiting bodies, that's what creates the conidiospores or sporangiospores. And then once they start releasing spores all over, that's a danger to our patient. So here's the aerial hyphae where these conidiospores and sporangiospores will bud off of those hyphae. So we talked about spores. Here's sporangiospores. It sounds like spore. It's a spore inside of a large spore, a bunch of spores inside of a spore. And then conidiospores are the naked spores that will just disseminate. So once you don't have to know this yet, we'll figure this out later in the lab, but here's rhizopus. That's characteristic of one of our pathogenic fungi that have sporangiospores. And then conidiospores are aspergillus or penicillium are our two main ones that we worry about. So here's what they look like on a plate. They look fuzzy. Here's under a microscope. They look like spaghetti meatballs. Here's the dimorphic. So they'll be in their pathogenic form, which really, I mean, they are pathogenic either way, but they'll be at warmer temperatures. They'll have their own yeast structure, and they can have a mold structure when they're in colder temperatures, which we worry about transmission because now they can survive and thrive outside of the human body. Here's some examples of dimorphic fungi. We don't have to know those yet. Our only one that we have to know for the final is the Candida, because that's our no part. Okay, here's some terms. Is there anything else you guys want me to go over so far? Let me see. Oh, metabolism. Let's do that really quick. Okay, so yeasts are facultative, molds are strict aerobes. So which ones have a electron transport chain?
The molds.
Both. They both do.
Yeah, they both do.
Which ones create acids? Facultative yeasts create acids. So molds don't make any acids. What else do we need to know about that? So which ones have a mitochondria? Both. All eukaryotes have mitochondria. Okay, so we talked about CPE, so direct invasion is a problem because of the Spitzenkorper or yeast actually, because when they have pseudohyphae, they really can dig into tissues as well, but a lot of times they'll degrade the tissues around them. So direct invasion is always a problem with
And that's the digging?
fungi. Yeah, so any kind of digging or invading tissues of any kind.
Like the vegetative.
Mm-hmm. Yes, yeah. Vegetative hyphae it's like your patient has a very low chance of survival once the vegetative hyphae get into the lungs. But yeah, so we worry about that a lot. But yeast, depends on if they make pseudohyphae or not, but also if they're secreting enzymes that degrade the tissue around them, they can still get into the tissue. Yeah.
Were those Spitzenkorper, is that just for molds?
Just molds.
And then the pseudohyphae is kind of like their Spitzenkorper in a way?
Yeah, and they don't have a Spitzenkorper, so they just create spaghetti strands, but they're
How
not.
are they able to like dig into the tissue if they don't have that Spitzenkorper?
So because they secrete those digestive enzymes still, yeah, so they secrete digestive enzymes and then they just end up residing in the tissue. They don't really dig, but they do invade the tissue because they've degraded the tissue around them.
Okay.
This is yeasts.
Yeasts. Yeah. Yeah.
Besides the Spitzenkorper, are there any other major differences between the mold and yeast for structure? It's basically that.
Yeah, yeast don't create spores. Yeah, not really.
Okay.
Yeah, good question.
Are the Spitzenkorper a fermentation byproduct?
No, they're like really hard crystals, and then they have enzymes too. So they're like little rocks almost, and then they have enzymes. So those enzymes and the rocks will it's like digging in a rock with a spoon or something like that. Degrading it. So the toxic enzymes are one of the cytopathic effects. They're not really considered mycotoxins because mycotoxins are extremely potent. And even if you treat your patient with an antifungal, they have a mycotoxin problem. So that's a whole nother beast. A lot of times they'll do like a chelator. They'll give them like charcoal or something like that so that that'll chelate the toxin. Yeah.
Can you talk really quick about the 5' and 3' ends of the caps?
Oh yes. Mm-hmm. Yeah. So in a traditional, so let's just give humans as an example. This is like a human epithelial cell. Here's the nucleus. And so during transcription, the DNA will be transcribed into RNA. So then that RNA, so DNA, it's linear DNA, it'll open up and it'll be transcribed into RNA. So then you have an RNA piece, and that RNA piece will be spliced. So this is still in the nucleus. And they're called spliceosomes. They'll splice out those introns. And these are exons. Now you have mRNA after it's added a 5' cap and a poly A tail. And now it can leave the nucleus.
Okay, so just know the poly A tail and 5' cap are added for stabilization.
Yep, and it's spliced. Now it can leave the nucleus. Now it's safe to leave the nucleus. It has to go through splicing and cap and tail before it
Do
can
we
leave
have
the
an idea
nucleus.
what the purpose of that 5' cap is?
Yeah, it is to help it get out of the nucleus. So the nucleus is extra protective, more so even than the cytoplasmic membrane. The nuclear envelope, the nuclear membrane is really selective about what lets in and out. I'm sorry, what?
Doesn't the nucleus have pores in it that
It
allows?
does have pores in it, yes, but it's very selective about what it lets in and out.
Okay.
So it has to have that cap on it to be able to leave exit.
Okay.
Yeah. So both yeasts and molds can sexually and asexually reproduce. Fragmentation is asexual reproduction where it just fragments. Fertilization is when DNA is shared from an antheridium to an oogonium. So the male to the female, its DNA is disseminated. And then fusion is when they can come together, when the male and female parts of the hyphae will come together to form this environmentally resilient structure. And we worry about that clinically because those are hardy structures that you can't get pharmaceuticals into. Does anybody have any more questions? Feeling pretty good? No? And we have our lab practical. We'll have about 30 minutes for the lab practical to, if you guys want to just do like crazy note cards together, you guys can use this room. I'm gonna go and just do one more round of setup, make sure all the microscopes are set up properly and everything. So you guys will have 30 or so minutes in here. So you guys can stay in here, and then you guys can use the board if you want to and write out like INVIC and write out like some of our media and selective and differential and go through some of the tougher ones.